Hello and welcome to my protein project video. The protein is alpha epithelial sodium channel, known as alpha ENAC. The normal function of the protein is that it facilitates sodium ion reabsorption across the apical membranes of epithelia in the distal, infrone, respiratory, and reproductive tracts, and exocrine glands. The categorization of the function of the protein is transport because it allows certain molecules or ions to diffuse across the membrane. And in our case, those ions are sodium. The protein is located in the apical membrane of polarized epithelial cells. In particular, those epithelial cells in kidneys, lungs, skin, the colon, and sweat glands, as well as the reproductive tracts of both females and males. It also plays a main role in taste re- perception. When the protein is mutated, the activity of the function increases because the mutation of the protein causes it to fasten the transport or the current of sodium ion reabsorption. And the reason to that is that the mutation increases the number of ENAC channels at the surface of the epithelial cell and becomes hydrophobic. Now, the 3D model of the normal ENAC channel represents three subunits alpha, beta, and gamma on the cytosol of the epithelial cell. Now you would see those N, like those tails, have N next to them, as well as for beta and gamma. That N is the nitrogen terminus, indicating an amino group on this side, and the C represents the carbon terminus, which is the carboxyl group. Now, the model of the mutated channel represents the C479R mutation on the alpha because it is somewhat pointed towards the outside of the cell, unlike beta and gamma where they're already inside. Also, we have the sodium ion reabsorption being fastened. So, as mentioned before, the mutation increases uh, the ENAC channel activity as the current of the transported sodium increases, causing an increase in cell surface density of the channel and protein truncations that induce abnormal operation of certain key components of the ion channel. So you see right here that the subunits are tilted towards each other right here, unlike where we have it right here a little bit far, or like there's some space like between them, because we have the the fast transport of sodium ions. That's why they're tilted and closer to each other. The normal protein has the cysteine as the amino acid, which is hydrophilic, That's why the transport of sodium ions is not fast and it's not slow. It's just normal. And the main functional group of cysteine cysteine is sulfhydryl group. Whereas for the mutated protein, the amino acid is arginine, which is polar and hydrophobic, uh, causing the rapid transport of sodium ions. And arginine has two functional groups, carboxyl groups and amino groups. And that's where the terminus of nitrogen and terminus of carbon play a main role in arginine and in the mutated protein. Now, there were three limitations faced as the model was being created. The first one is that the model only represented the subunits and not the full protein. Secondly, it did not include the effect of amiloride in the normal model, where the potassium sodium exchange would occur around the protein or in the cell. And lastly, the model did not have the binding sites around the subunits, where the subunits would bend with another molecule in the protein or in the cell. And that was it for the protein alpha epithelial sodium channel. Thank you for watching.